Hi, everyone. Thank you for joining the fifth webinar in our series on the use of NAMs in risk assessment. This series is being co-organized by the EPA, the Physicians Committee for Responsible Medicine, and the PETA International Science Consortium. My name is Amy Klippinger. I direct the PETA International Science Consortium, and I'll be co-moderating today's webinar with Christy Sullivan, who is the Vice President of Research Policy at the Physicians Committee for Responsible Medicine. We have two speakers today presenting on CATMoS, development and use of the Collaborative Acute Toxicity Modeling Suite. And I'll introduce Drs. Nicole Kleinstreuer and Kamel Mansouri in just a minute, but before I do that, I'd like to point out that the slides from today's webinar can be found online at the link on the screen, and a recording. Both will be posted in the next few days. The slides and a recording of the first 4 webinars in this series are also available at this same link. There will be time for questions for both speakers at the end of the second presentation. Everyone is on mute, but you can ask a question by typing it into the question section of your GoToWebinar toolbar, which should be on the right-hand side of your screen. So speaking first today will be Dr. Nicole Kleinstreuer. Nicole is the Acting Director of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, or NICETM, where she leads domestic and international efforts to develop novel testing and analysis strategies that provide more rapid, mechanistic, and human-relevant predictions of potential environmental chemical hazards. She has a secondary appointment in the NIEHS Division of Intramural Research Biostatistics and Computational Biology branch, and adjunct faculty positions in the Yale School of Public Health and the Eshelman School of Pharmacy at UNC Chapel Hill. Then after Nicole, Dr. Kamel Mansouri from Integrated Laboratory Systems will be speaking. So Kamel is leading the computational chemistry efforts at Integrated Laboratory Systems, supporting the we'll say NICETM. He is well known for his international collaborations and leading consortiums of renowned scientists in the field of QSARs and computational toxicology. Nicole, whenever you're ready, you can go ahead and start.
Okay, great. You can hear me just fine?
Yes.
And you can see the title slide?
Yes.
Okay, wonderful. Well, thank you so much, Amy and Christy, and to the PETA International Science Consortium and Physicians Committee for Responsible Medicine. Also, thank you very much to EPA, to the joint organizers of this webinar. Very excited to be talking to you today about CATMoS, the Collaborative Acute Toxicity Modeling Suite. And I'm going to go through an introduction of the project. I'm going to talk to you about the scope of the project, how these data are typically generated for regulatory use, which endpoints are important to various regulatory agencies, how the data are used, and then how we went about this project of compiling a training data set that was large enough and robust enough that we felt that with our international collaborators that were really all the modeling groups in the world that are at the forefront of leading quantitative structure-activity relationship modeling, where we felt we had enough training data to really confidently be able to build a robust predictive model to hopefully at some point replace the animal test for acute oral LD50. So I think everyone understands the concept of why we need alternative approaches, not only from an animal welfare standpoint, but simply from a resources standpoint as well. So there are far too many chemicals in the environment that people are exposed to actually test with standard animal-based methods. And so that leads us down this path of asking whether or not we can actually build predictive models that will relate the structures of those chemicals, so their molecular features or their physicochemical properties, to a toxicity endpoint. In this case, the toxicity endpoint that we're interested in is acute systemic toxicity, in particular via the oral exposure route. So we did this project through the auspices of the ICVAM Acute Toxicity Work Group. For those of you who may not be familiar, ICVAM is the Interagency Coordinating Committee on the Validation of Alternative Methods. It's a collection of U.S. federal agencies, representatives from U.S. federal agencies that either require or consider safety testing data. EPA is certainly one of our strongest partners on ICFAM. And ICFAM has a number of different work groups that are targeted towards specific types of toxicities or specific types of methods or approaches to try and answer regulatory needs and decision-making problems. So in this case, we have an acute toxicity working group, and one of the first things that that work group did was to try and identify what are the different decision contexts and data needs and data uses and requirements across all these different U.S. federal agencies for acute systemic toxicity data. So the rat oral LD50 test is a fairly simple test. You feed a group of rats increasing concentrations of a substance until you see 50% mortality in that group, and that's what you call the lethal dose at which 50% of the population suffers mortality, so that's the LD50. And I very naively, when we started this project, thought that, oh, that's just informative for things like accidental exposures, poisoning type cases. But as it turns out, this one value, this LD50 value for a particular substance, has a vast myriad of uses across different agencies and in different regulatory decision-making contexts. So it's used by the EPA to define their hazard categories, which you see here on the bottom left-hand corner of the screen. And those hazard categories inform things like classification and labeling of these compounds, whether or not you have to use child-resistant packaging, whether or not certain compounds are able to be used in products in schools, for example. Other agencies like OSHA or the Department of Transportation rely on the globally harmonized system of classification and labeling. So they have their own categories, which are similar to the EPA categories, but have slightly different cutoffs in the middle there that you can see. And those are used for things like transportation of goods across state lines and labeling packages appropriately. Other agencies like the Department of Defense or the Consumer Product Safety Commission also use LD50 values, but from different perspectives, like trying to understand whether or not a substance may be highly toxic. So there may be a necessary immediate threat response in response to exposure to a highly toxic substance, or from the perspective of looking at green chemistry design, so being able to identify non-toxic substances, so substances that have very high LD50 values, so in the thousands of milligram per kilogram per day, and not being toxic at those doses. And then, of course, other agencies are also interested in really knowing not just whether something falls into a particular category, but what is the actual quantitative numerical value of that LD50 so that they can do things like inform quantitative risk assessment decision-making processes. And so when we queried all of these different agencies and got all these different answers back, we decided that we would put this to our group of collaborators as the objective to try and build models for each of these different types of applications. So we have binary models where you're trying to predict whether or not a compound is very toxic compared to everything else. So it has an LD50 below 50 mg per kg per day. Trying to predict whether a compound is non-toxic, so it has an LD50 above 2,000 mg per kg per day, and differentiate that from everything else. Trying to build categorical models that correspond to those different classification and labeling schemes, so the EPA categories, the GHS categories, so multi-class, 4-class, or 5-class models. And then, of course, trying to predict those actual quantitative LD50 values by building regression models that will predict a continuous endpoint of that LD50. So with our partners at EPA, we did a substantive data curation exercise where we went and pulled all of the available information in various databases, such as the hazardous substances databases, registration submission packages to EPA, to ECHA, and compiled over 34,000 LD50 values for over 16,000 chemicals. So obviously there's a subset of chemicals that have multiple LD50 values. So they had multiple independent studies, and that's useful for a different purpose, and I'll touch upon that a little bit later. But then what we had to do is we had to go in and we had to prepare this data set for modeling purposes. So we had to make sure that all of the chemicals that we were using to build and train our predictive models had defined structures and that they were really appropriate for building quantitative structure-activity relationship models. And to do that, we relied on work that's been done over the span of several years, building off of groundwork laid by Alex Troche's group and Denis Forsch, and then relying on a workflow that was designed by Kamel and is implemented in the KNIME pipeline system. And so you can see a sort of a snapshot of that here that is really designed to standardize those structures and minimize the differences between the structures used for predictions and create a very clean data set that ensures that all of the modeling groups are using the same structures for both training and testing their models. And so you can see the different steps outlined here in terms of how we remove inorganics and ill-defined structures and mixtures and normalize the tautomers, remove duplicates, etc. And this is a free open source workflow that is available upon request. So then after we went through this QSAR-ready structure standardization, we ended up with almost 12,000 chemicals in our final inventory that had rat oral acute toxicity, so LD50 values associated with them. Then we went through pretty standard, good modeling practices of splitting that data set into training and evaluation or test set. So we did 75% training and 25% test. This was done semi-randomly, by which I mean that it was mostly random, but we did ensure that we had similar distributions for different types of values. So making sure that we had a similar distribution of LD50 values, because we wouldn't want to have all of the very potent, highly toxic, category 1 chemicals by coincidence falling into the training set and having none of them in the test set, for example. Also making sure that we had similar distribution of sources across the chemical structures to make sure that if there were any sort of errors that may have come from different databases, that they would be equally represented. Anything that we couldn't control for would be equally represented across both the training set and the evaluation set. So then we established an additional set that we called the prediction set. And so these are chemicals which the vast majority of them we don't know the LD50 value for, so they don't have any associated rat acute oral systemic toxicity studies that we could find. But they are chemicals that have been prioritized in terms of their interest and their relevance to different regulatory programs. So, much of the TSCA inventory is on this list. The list of prioritized substances for the endocrine disruptor screening program, various lists that we derive from the EPA CompTox chemicals dashboard as far as substances that are on the market and may have significant potential for human exposure. The ToxCast and Tox21 chemical libraries, which also cover a lot of these objectives. And so after, again, running through that pipeline to standardize the structures to make them ready for QSAR modeling applications, we had around 48,000 structures. Hidden within that prediction set were the almost 3,000 structures that we had held out as that evaluation set. So if you think about it, what happens is we give about 9,000 chemicals to the participating modeling groups with associated LD50 values, and those 9,000 chemicals are the training set. So everybody uses the same training set to train their models, but everybody's using slightly different algorithms and slightly different feature sets and molecular descriptors. They have their favorite ways of approaching QSAR modeling, but they're all using the same training set. And then there's about a 3,000 chemical evaluation set that's hidden within this 48,000 prediction set. So we just gave them the structures for the 48,000 prediction set and said, please give us not only the predictive performance on the training set and the cross-validated evaluation on the training set, but also please give us the predictions for all 48,000 chemicals in the prediction set of which we knew the identity and the quote-unquote correct answer for 3,000 of those chemicals, and we could use those as the external validation or evaluation set. So we had 35 different groups participating from around the world. So you can see a map here of all the different locales. So obviously very heavy representation from the US and Europe, but we also had some from South America and from Asia as well. And we had representation from various sectors covering academia, industry, government, etc. In total, we received 139 models from the 35 different groups. And that's because we didn't really set any restrictions. We just said, if you want to participate in this global collaboration, you just have to submit at least one model. So pretty much almost every group tried the binary modeling approach, which is usually considered to be the easiest type of model to build. So distinguishing non-toxic chemicals from everything else or very toxic from everything else. And then it kind of stepped down from there. Some groups submitted 5 different models. So they had 2 binary, 2 multi-class, and 1 regression model, and others just submitted various subsets. But in total, we had almost 140 models that were submitted. Then we went through both a qualitative and a quantitative evaluation procedure. So we, in order to be consistent with the OECD, so the Organization for Economic Cooperation and Development, principles on validation of QSAR models, we looked for these various qualitative factors around documentation and availability of the code and the unambiguous algorithm, etc. We also did a quantitative evaluation where we looked at cross-validated statistics on the training set, and then also, as I mentioned, having this sort of holdout evaluation set where we were able to evaluate performance on the external validation set. And we looked for robustness of the model, so the balance between the performance on the training set and the evaluation set to ensure that models weren't overfit to the training set. And you can see here some of the metrics on how we actually evaluated those categorical and those continuous models. So here's kind of a snapshot of the coverage and the concordance of the models. So one really, really nice attribute of taking this like global crowdsourcing type approach is that, as I mentioned, everybody has their own method that they like to go about building QSAR models, and along with those comes things like the applicability domain of the models. And so not every model is able to individually create a prediction for every single chemical. But if you combine all of those models together, you do get multiple predictions for every single chemical in the prediction set. And what we find in general is that the more models agree on a particular prediction, the more likely that prediction is to be correct. And so that's what's reflected in that predictions concordance. And as expected, so the binary hazard models agreed more often, and then the more difficult endpoints like the multi-class models and the continuous models had sort of more of a spread across all the different modeling approaches as far as their concordance across the prediction set chemicals. So then we took the steps of creating a consensus model. So we had all of these different models for these different endpoints, and to combine those models, we used a weighted average or majority rule approach, and that gave individually for each of those different types of models. So for very toxic, we combined 32 models into a consensus very toxic model. For the EPA categories, we combined 26 models into a consensus EPA categorical model. So that gives us a consensus model for each of the 5 endpoints. But as I'll show you and as I'll step through in the next few slides, there are occasions where those consensus models may give slightly different predictions for an individual chemical. And so then we took a weight of evidence approach to combine all 5 of those consensus models into consistent predictions, so a true overarching consensus model that represented all 140 models that were submitted to us. And I'll walk you through an example of that here. So for molecule X, we had these 5 different consensus models representing the 5 different endpoints. For this particular chemical, the very toxic consensus model predicted that it would, so it predicted a negative, so it's not predicted to be a very toxic compound. So it's not predicted to have an LD50 below 50 mg per kg per day. So that's what's reflected in that top row. So this orange bar, this orange range is the LD50 span that would be predicted by the fact that it's not supposed to be very toxic. It's also not predicted to be non-toxic. So it would not be predicted to have an LD50 above 2000. So this is the range predicted by that non-toxic model. So just by those first 2 models, we know that it's somewhere in here in this middle range between 50 and 2000. Then we step down and we look at the EPA categories. So this was predicted as an EPA category 2, which narrows it down to it's somewhere between 50 and 500. It was also predicted to be a GHS category 3, which narrows it down to be between 50 and 300. However, the LD50 prediction, and this is the log LD50 value that you see at the top of the screen here, and if you transform that LD50 prediction was 316 mg per kg per day. So it's actually just above that threshold. So it's inconsistent with the GHS prediction. So the GHS prediction was 50 to 300. The quantitative LD50 prediction was 316, just above that threshold. But as I told you earlier, we also had a subset of over 1,000 chemicals where we had multiple independent LD50 studies or LD50 values from independent studies for individual chemicals. And so then we could do something where we could actually query what is the variability of that animal study. If I ran an LD50 test and got, a number X, would I expect to get that exact same number again if I ran another LD50 study in a different lab at a different time, but on that same chemical? And as you might anticipate, that the answer to that is no. You would never expect to get the exact same number twice. There's got to be some inherent variability, some confidence interval that you can apply. So without getting too much into the details, and we're preparing a manuscript on this analysis right now, we bootstrapped through the standard deviations that we had for all the chemicals where we had multiple LD50 values from independent studies, and we were able to derive a variability range for that quantitative LD50 value that reflects the variability that we see when multiple independent studies are performed. And so that's about plus or minus 0.3 log units. So then when we apply that confidence interval to that quantitative LD50 value, we get a new range for that LD50 prediction. And that puts it in those other 2 categories. So now we have a consistent weight of evidence schema that we can apply here. So if you basically just add up all the boxes and say, okay, where is there the highest level of evidence that this chemical should fall into a particular category based on all of these different consensus models, you see that it falls into this middle category right here. So between 50 and 300, and that's consistent across all of the different models that we have built here. And so that's that winning bin. And so then the question becomes, well, how then do we adjust that quantitative LD50 value to reflect that weight of evidence approach? And we looked at a number of different types of mathematical formula to do that. And as it turned out, the one that gave the most consistent results was just looking at the average of that lower confidence interval that was applied to that quantitative prediction and the upper threshold of that bin that's consistent with all of the other model predictions. So here that would give you an LD50 of 230 mg per kg. And so creating a consistent call that is consistent with all of the other models leads you to a log LD50 of 2.36. So hopefully that's pretty clear to everybody, and if not, we can answer some detailed questions at the end. So now we have this weight of evidence, overarching, consistent model that combines all of the consensus models that were built independently by independent groups for these different types of endpoints. So it's an incredibly powerful approach. So how does it perform? So we looked at the performance assessment in comparison to the performance of the in vivo animal test. Now, because we don't have human data to compare the animal data to really know how well the animals are actually performing and predicting acute oral systemic toxicity, as a surrogate, we used the performance of the in vivo animal test at replicating itself. So again, looking at this subset of chemicals where we had independently run studies, how often do they agree with one another when you look at the criteria for being able to predict whether something is very toxic or non-toxic or falls into each of these categories. And so that's what you see here in this in vivo balanced accuracy row at the bottom. So that's how often do replicate in vivo studies give you the same answer on the same chemical. And so that's a decent, I think, surrogate for establishing expectations for performance of a predictive model. You wouldn't expect that a predictive model would do better than the existing in vivo reference study at replicating itself. And so you can see that obviously the statistics are a little higher on the training set than they are on the evaluation sets, but by and large, very similar performance between the balanced accuracies of the QSAR consensus models when compared to the balanced accuracies of the in vivo animal test. And so that's what's shown up here for the categorical endpoints. And then down here, you have the R-squared value and the RMSE, the root mean squared error for the LD50 values, the quantitative ones. So then how do we extend it? So as I already shared with you, we had 12,000 chemicals between our training set and our evaluation set. We had another 48,000 chemicals that we asked the participants to provide us predictions on. So we have a weight of evidence consensus prediction for the 12,000 chemicals where we already had the data and another 45,000 chemicals from the prediction set, but there are hundreds of thousands more chemicals in the chemical universe. And so how do we generate consensus predictions, extended predictions that reflect those consensus models on all of those additional chemicals? Because some of our participants, they come from they have proprietary software that they were using to apply to this problem. And so, or there's, complex code that was submitted to us. And so it's not necessarily reasonable that we should be expected to run all 139 models in-house, combine them into a consensus for the hundreds of thousands of chemicals in the chemical universe. So what we've done here is we've created a weighted read across, so a nearest neighbor approach to extend the CATMoS predictions from that set of around 60,000 or so that we have all the consensus model predictions for, and extend that to the hundreds of thousands of additional chemicals that we would like to see the predictions for. And this is just a simple weighted K nearest neighbors. The nearest neighbors are determined based on the selected descriptors for the endpoint in question here. So based on the descriptors that were the most important in establishing the consensus model for acute oral toxicity. So this just shows you, this is basically summarizing what I just said, but if you're interested in a summary, a high-level summary of how we got to this point, and in particular the workshop that we had to establish sort of these ground rules and establish the collaboration, that was published at the end of 2018. You can see the citation there. So we are partnering with several of our ICVAM agency stakeholders and acute tox working group partners to try and evaluate these predictions. So obviously each agency has particular regions of the chemical space that they care more or less about. And so we ask them to nominate substances to us that either they don't know the LD50 for and they would be interested in seeing the predictions, or maybe they know the LD50 but it's confidential data, so it wasn't part of our training set, part of the data that we curated. And so these are the numbers of substances that various agencies have nominated to us and that we have produced CATMoS predictions on, and we're working hand in hand with each of these agencies, in particular with EPA, to evaluate how well CATMoS does and potentially optimize some of the predictions based on the chemical space of interest. soon you will be able to find all of these predictions on the EPA's CompTox chemicals dashboard and on our integrated chemical environment page where we host all of NICEDM and ICVAM's data. So I just want to say a quick thank you to all of the folks that were involved in this international collaborative effort. Certainly there's not enough space on this slide to list all of the groups that submitted models to this, but it was just a really tremendous activity across the globe, and it was really an exciting thing to be part of, and it's a model that we hope to carry forward to other endpoints where we have sufficiently robust training data. And so here is where I'm going to turn it over to Kamel to actually take us through the process of how to run these predictions. So how to download Opera, how to run it, and how to derive these acute toxicity CATMoS predictions and any other predictions that you might be interested in that are contained in Opera. So thanks very much, and I'll turn it over to Kamel now.
Thanks a lot, Nicole. Do you all see the screen with the GitHub page?
Yes.
Okay, perfect. So thanks again, Nicole, and thanks everybody for joining, and thanks to the organizers of this webinar. So as Nicole mentioned, the CATMoS model and also other models are implemented in Opera, which is a free and open source software or suite of QSAR models that anyone can use. Take a look at the code, use it for different reasons, or just download it and apply it. The predictions, as Nicole mentioned, also exist on the EPA dashboard and on ICE, and CATMoS will be there very soon, CATMoS predictions. But to use the app to run the models, this is, I'll walk through like the different steps, the main steps to use it, and how to interpret the results. And at the end, if anyone has any questions, we'll be happy to answer them. So if you click on the link that Nicole showed in the last slide of the GitHub page for Opera, you will land on this page right here. So for anyone who's not used to using GitHub, the first page is mainly about the source code. So unless you're a developer and you want to take a look at the code, then you need this first page and you can download the code or look at it. Otherwise, I just want to mention a couple things about this first page, which is, first of all, if you want to learn a little bit more about the history of the app and how it was built and the different QSAR models and everything, these are some links, literature, manuscripts and papers that were published about Opera, and here's the list of models, but we will get back to that later. So the most important thing right here in this first page is this install guide. So I invite everyone who wants to use it to first take a look at this. You can take a look at it here right away on the online, or just download it. So once you download it, you will have this PDF file that walks you through the different steps of installing and running it for the first time. And a lot of information, some of it Nicole already mentioned, and some will go through right now. So as you can see also in the first page of this PDF, I'll leave this aside right here and go back to the main page. It says you need to go to the releases. So this is the second step, and most important step is to go here to the releases to see the latest release. So here it might say 2018, but that's just when we started this new versioning of Opera. So don't worry about that. As long as it says here latest release, then you're good. So then this is the version of Opera, and then you have different choices to install, because there are different versions It's all version 2.5, but different uses. So as you'll see in the PDF file, we have a version for Linux, a version for Windows, and there's for each one of those two, there's a command line and there's a graphical interface. So right here today for CATMoS, we will be focusing on the Windows version, the graphical interface. So that's what it's here. And just note that all models will, once you install it, they will run locally on your desktop or computer. You can disconnect your computer and it will run. It does not connect to the internet. It does not post anything online. It does not even look for new versions. You have to watch here the page to get updates, or maybe the feed, the NICERM feed, and the NICERM page on the NTP website to get notified of new versions. And we do that to make sure that anything you run on the app that you download and install, it runs locally. So those are the different files. Then you go a little bit down, you'll see all the different updates through the versions. So the main updates in version 2.5 are that the standardization workflow was updated a little bit, the one that Nicole mentioned, and we now have a more robust workflow that is based on the 900,000 or around 900,000 chemical structures from DSS Tox database that is on the EPA dashboard. So you can also download and run this workflow, the 9 workflow that Nicole mentioned earlier, and it's also available on GitHub. So in addition to that, we also have in Opera, when you download it, you have all the structures of those 900,000 chemicals from DSS Tox. So you can only, if you want, and if you don't have the structure of the chemicals that you want to predict and only have one of these IDs like SRN or DSS Tox ID or something, you can use those also to make predictions. But if you have the structures and you want to use your own structures, you can also do that. And more about that later. So, and we also added new data for certain models and improved some others and fixed a couple bugs from the previous version and improved the speed. So the other thing I want to mention is the tool requirements. So this is what you need to have. It's not a lot, and we tested it on different versions for Windows and Linux, but up to a certain date. Also, you will need, of course, a structure or an ID to run your chemicals. And there are some limitations that are not related to Opera itself, but to the molecular descriptor calculators that we're using in Opera that are CDK and PADEL. So those, especially CDK, has the limitation on the size of the chemicals or the number of heavy atoms, not including hydrogens. And also it does not take any inorganics or mixtures or nanomaterials. And then you have the list of models, but you will have those later on. So as I said, what we want to focus on is the Windows graphical user interface version, which is very easy to locate, the last one right here, and it's only 138 megabytes. So you download the zip file. It takes only a moment, but I already have it ready here. And when you unzip it, this is what you will get. So again, you will find the PDF. If you didn't look at it on the GitHub page, you will find it again in the zip file. And then you will find the list of models that I invite the users to take a look at. If they're, even if they're interested in one model, you can take a look and see the other models that are available. And you also see the versions, the units of the different models, and also for some of the existing models, you also can find the QMRF report, which is the European REACH or GRC way of documenting and saving QSR models and their report ID that you can take a look online. So that's the list of models. And then you have a couple sample files, input files to start with to test the app. And of course, mainly the installer. So the installation is really simple. It only requires admin rights for the first time to run it. So if you're not admin on your computer, you can ask your IT department to install it for you for the first time. And once you have that version or previous version, then the installation goes like really fast because it will detect the previous version and it will only update or change the files that change. It will not reinstall everything from scratch. So this is where you start, and it's really simple. After that, you just click next, and it will install. If it's the first time, you add a shortcut on the desktop to double click on it to make it easier. Otherwise, if you're just updating, you just click next, and install. And it only takes a moment to install once you have a previous version, because only a few things will change every time, and that's it. It's installed. You have it on your computer. So here I have, I forgot to mention that the other thing, other than the command line and the graphical interface, in this latest release, we also have a command line version that runs in parallel, meaning that for users who use it for thousands and thousands of chemicals, it might be useful. And I personally used it to make the predictions for the 900,000 chemicals from DSS stocks to populate the EPA dashboard and the ICE, NTP ICE dashboard as well. So this is something that advanced users or users who have heavy files with thousands of chemicals, they can use in command line, and they can even script it and include it and implement it or embed it in their softwares or scripts. So I have here all three versions installed for Windows, but as I said, we're only going to focus on the graphical interface version. So it uploads the models, it loads the graphical interface, and then this is how it looks like. So as you can see, you have different compartments of the of Opera. You have here the models, you have the output options, the results and summary, the button to calculate the models, but of course the input and output. So for each one of these, you have this information button that you can click and get information on what you can expect. So the input files can be in different formats. It has to be a structure, and its structure, it can be in SMILES, in SDF or MOL file, and those are guided again by the different cheminformatics tools that are available that we're using in Opera, but also these are in like universal structured formats. So it has to be in certain way for the descriptor calculators to be able to parse it, interpret it, and run it to get the molecular descriptors, which is the first step that Opera runs, and then running the models. The other option, as I mentioned earlier, is to have a text file containing the IDs if you don't have the structure ready, or if you want Opera to get the right structure, the QSR ready structure for you. So that's the input. The output is also having options. You can have it in CSV, and then it will be one row per chemical no matter how many models you run, or a .txt, and then they will be listed by chemical for all the models. And it depends on the use. The second version, I'll also show it later, the text file, that it can be converted to PDF more easily for people who want the results to look like a report or something. But most users, I guess, they're going to use the CSV, which is 1 row per chemical. And then you have the models. So as I showed earlier in the text file, you have the models, the abbreviations here. Each model is abbreviated, but if you want to know what it means, what's this model, what's BCF, then you come here and then log BCF. That's the fish bioconcentration factor, and the version. The version is not always the same as the Opera version itself. So the interface or the tool itself can be updated to fix certain bugs or to add new features without necessarily changing the versions of the models. The models might remain the same. And the only reason that now you have them the same as the version is because we updated the structures, as I mentioned earlier, and added data and so on. So, and you always have some references to know more. The output options. So let's say you have you want to run different models and you have 1,000 chemicals and you don't want all of them for all models to be in the same file. It will be too heavy to handle. Then you can click here and have them in separate files. You can add the experimental values if you have the CATRN or an ID in the file, and then the nearest neighbors and so on. So, and this part right here, it will summarize the main steps of running the tool. It will tell you also how much time, and if there are errors or issues with the structures, it will be also mentioned here. And there will be, if there is an error or warning, or like, for example, if you exceed the limitation on the size or something, there will be a pop-up window that says that. If you click here, you always get the version. And also, like, if it's a beta or, you know, and the support. So, this is great. It's still an option coming soon, which is running that workflow that we mentioned earlier on your input chemicals. But let's start with the samples that we have. So when you click on input, it will get to it will let you upload the file that has the chemical structures. So as I mentioned, it can be in a SMILES or an SDF, and you click on the file that contains your structures, and I'll show you how it looks like later, and then you click load. So it immediately calculated how many structures it has in that file, and it also automatically populated the output file as a CSV. But of course, you can change it to .txt here, or you can change the name, or you can even change the location of the output if you don't like it. So here, I'm going to change it to SMI just to show you how it looks. And then it will run in that same location where you have the output. So that's oh, sorry. I accidentally closed it. So it will run in here, and that's where you're gonna get your output. SMI. Okay, then you select which model and you run calculate. So you can select as many models as you want. It will show you the progress and what it's doing right now. Like right now, it's calculating the descriptors, the 2D descriptors, and it only calculates the descriptors that are necessary for the model or the models that you selected. And then just because CATMoS is one of our biggest models, it requires descriptors from both PADEL and CDK, and then it's running the model right now, and then it will write the output file, and that's it. So it calculated predictions for 50 chemicals in 42 seconds, and it gives you the details here for each one of the different steps. So total 42 seconds, and how long it took. So it took only 7 seconds to run the model itself. Most of it goes into the descriptor calculation. So let's take a look at the output file right here. So just gonna make sure that it's run correctly. So you have, and we will go back later on to interpret the results, but just to show you that this is the output that was written right now. You have the ID that you have in your input file, and then the predictions. So let's leave it aside for a second and go back to see the input file. So the input itself is like this. So this is the file, the .smi file, which is mainly the SMILE structures, and then followed by a tab and the CAS number. So the CAS number is an optional, or any other ID. You can put any other ID, and it's optional. You can also have a SMILE file with just the structures. The important thing is, as in here, it will say that the important is that no headers. It doesn't need to know. It knows that this is SMILES and this is an ID, and that's what it will use to identify the chemicals later on. Otherwise, it will generate an automatic ID for each one of them, and it will keep the same order for you. So, and no empty lines, no commas. So, and you can read online about what's a SMILES file, but basically that's it. You just copy the SMILES, and if you have an ID, you can put it there. The SDF is a little bit more complicated. You don't need to use it if you don't want to, unless unless you're using some other chemoinformatic tool to create it, but you don't have to. The important thing, and all these details are mentioned in the PDF file, by the way. So you have all of them right here. So these are the different input files. What's important for each input file, it's all in the PDF. But the version of the PDF format, it has to be V2000. You can put as many IDs as you want for an SDF, and you can put as many chemicals as you want as well. So the only difference between this and a SMILES is that it includes the coordinates. So this is a 2D SDF file, and it has the coordinates of the atoms. But that's it for now. Let's say you don't have the SMILES, and you want to create a SMILES file and run the prediction by yourself. So let's say you have this file right here that it contains the SMILES, the CAS, or some other ID, and you want to create a SMILES file. So as I mentioned earlier, the most important thing is that you don't take the headers, you don't take the title. You just copy paste them, either the SMILES alone, and you go to a text editor. I recommend Notepad++. You paste them here. That's a SMILES file. You only need to save it as text, and then you call it yes, SMI. And it has to have .smi as an extension. That's it. That's a SMILES file. If you want to add an ID that can be a CAS or just an integer or any other ID, then you take both of them They have to be tab delimited in the text file, but that happens automatically when you copy from Excel to a text file. It will be automatically tab delimited. The important thing is that you always need to have the structures in the first column. So if you have them inverted, you have to copy paste them and then have the smiles in the first column, and then you paste it right here. And that's it. You have 50 smiles with their IDs. You save it, and now you can use it in Opera. This is our smiles file. And you can use it to run the model, or any model, by the way. And the other thing is that if you, and this is just a Windows thing, it's not an Opera thing. If you have the output file open, Windows doesn't let you edit it using any other tool, so you have to close it, or just change the output name right here. If you change it, then you're safe. You're not going to overwrite an existing file. But if you want to overwrite it on purpose, then you just need to close it, and that's it. So it ran the models, and you have them here. So this is our file, and it contains the same predictions. The ID you see here, it uses the IDs to identify the chemicals. And if you don't use, let's say you go back right here and just to test, you don't have the CAS numbers, and you only have the smiles. Save it. We need to close the file. Don't save this. You don't need to keep that. Run it again, and it will generate automatic IDs for those structures.
So Kamel, I'm just a little cognizant of time because we're almost at the end of the hour. So maybe in the last minute or so, you could take folks through maybe another example with the 20 high priority TSCA chemicals, and you could highlight the output options as far as being able to see nearest neighbors and things like that.
Sure. Yeah. Don't we have it until 1:30? Sorry, 2:30 for 1 hour and a half, or that's it?
Amy, Christy?
We have scheduled it for an hour and a half.
Oh, okay, good.
Okay,
Sorry,
so we want to have some time for Q&A.
Yeah,
So,
okay, then I'll only take 5 minutes to run through a couple things that I think are important, and then we interpret the results and then leave time for questions. So so a couple last things. This is just the ID that I was just mentioning. It will automatically populate it with those generic IDs. One of the most important things, in my opinion, are the structures. So to get the most of the tool itself and the features that's offering, and the data that contains in the knowledge base of the chemical structures, I would recommend everyone that the first use, I mean, after running the samples, that if you have structures to predict, and if you have IDs, to just use the CAS numbers and let the app figure out which structures to use. So let's say you only have CAS numbers as in the previous files. You just copy paste them, save them as a .txt. You can also mix IDs. So let's say you don't have the CAT number for a chemical, but you only have the DSS tox ID. You can put it, or you only have the INSHI key. You can put it right there and let the app figure out which structure. And if it doesn't find any of them, it will let you know that, hey, I didn't find this one. And most importantly, for certain chemicals, when you have the smiles and it's a mixture and you don't know which part of the chemical you're going to use, by running it using the ID, it will know which part to use by itself. And sometimes you're trying to run an inorganic chemical, and then the app will let you know, hey, by the way, that ID is for an inorganic chemical, and you might not get, or even if you get prediction, it will most probably be outside of the applicability domain, because all models are built on organic chemicals. So for an example like this, where you only have the IDs, you can use that text file, which is right here, and then load it, and it contains 52 IDs, right? So we wanna run it and let the app figure out the right structure. So it's getting the structures right now based on those provided IDs. What it does is that it will create a smiles file for you to go back to later on. So you see, it already created the file that contains the chemical structures that it thinks are the right structures for those CAT numbers or IDs in general, and then it will go through the regular process of calculating the descriptors and then running the model for you. And just to show you later on, like right away, those that file contains 52 on purpose, and just to show you that when the chemical is inorganic, or that the CAT number it doesn't exist, or it's not part of the 900,000, most probably your chemicals will be there in the 900,000 structures that Opera has. But if it doesn't, it will let you know. So right now, when you use the IDs, it will use the same IDs to identify the predictions, and it will tell you how it found it. So it will know this is the Cataran, this is the DSSX ID, this is an INSHI key, and when it tries the smiles, that's the smiles file that it created, and the two that it did not find at the end, it says it will tell you exactly what happened. So this CAT, it doesn't exist. We don't have it. We don't know anything about it. And this one here is actually inorganic. It's an amide, and it's inorganic, so it will not run the predictions for those two. But let's try to see what the results mean. So for CATMoS, you have the 5 endpoints that Nicole mentioned in her presentation earlier. So you will have, and this is, by the way, for all the models, the last column. So you have the VT prediction, which is the very toxic, and then the non-toxic, and then the EPA category, GHS category, and then the CATMoS prediction, or the LD50 CATMoS prediction. And here it's already converted to milligram per kilogram. What we also have is the range. Remember earlier Nicole mentioned a plus minus 0.3 log value range, which is based on the in vivo data or repeatability of the in vivo data. So we use the same to give you the prediction, but also a range. And it's, of course, converted from the log value. So higher values, of course, will have a wider range. So these are specific to CATMoS. For all the other models, including CATMoS, you have the applicability domain, which is a binary column with 0 or 1. 1 it means inside the applicability domain, or 0 it fits outside. And then an AD index, which is the similarity to the 5 nearest neighbors. And then a confidence index, like how confident are we in that specific prediction, and that's also based on the similarity to the 5 nearest neighbors, and also to the predictions of the 5 nearest neighbors. And just to mention one thing that I think is important, on the EPA dashboard, you already have some of the OPRA models. So for example, for bisphenol A, it's just one of the examples that we use, you have the prediction for OPRA, but you also have the prediction report, which is all the details that you have in this file right here, including the applicability domain index, the 5 nearest neighbors, and the confidence index, and it tells you all about it. So this is just a graphical visualization of the same result, including the structures. In OPRA, you can also get the IDs of the nearest neighbors. You can also get the structures. Let me close this. Sorry, the experimental values, I clicked too soon. The experimental values for those chemicals. And by the way, you only get the experimental values if your input file contains a known ID, like a CAS number, a DSS tox ID, or a DSS tox compound ID. Then you can get the experimental values for those chemicals, because we only save the experimental values are per CAS, not per structure. So you get information about the nearest neighbors, their predicted values, and their experimental values, just like on the EPA dashboard right here. So this is general to the whole model, but this is specific to this specific prediction right here. So the predicted value, the applicability domain, the local applicability domain, and then the confidence level. It just finished. Let's go take a look. So now you have more stuff in the file. So you have the CAS numbers of the 5 nearest neighbors. You have their INSHI keys and DSS tox IDs. Maybe if you don't need those, you can hide them or delete them as you prefer. And then you have the experimental values and the predicted values for each one of the nearest neighbors. So if the nearest neighbor has an experimental value, it will be there. Otherwise, you'll get the predicted value, and you can compare. So based on this similarity index, it's very similar, and most of the time it's the same. If it's very highly similar, then it's the same as like you find it in the first nearest neighbor, and then you get what was already saved as a prediction for that specific chemical. Yeah, so I guess that's it. Oh, yeah, we can, we have right here the, I have it on the desktop, the list of the TASCA chemicals. And you can run it as well and get predictions for that TASCA chemical. And by the way, the file is where the file is. So yeah, I just put their CAS numbers as an input, and then let it get the structures, and it found, it's actually on the desktop. Okay, Windows again. Cannot open two files with the same name at the same time. Don't save it. And yeah, that's the other thing I wanted to mention, is that be careful with Excel on cast numbers. Certain cast numbers are converted to dates on Excel. So I usually use Excel just to see, not to save. Not to save cast numbers because they can be converted to dates, and then Opera will not understand what it means. It does not know that it's a cast number, or any other application for that matter. So these are the predictions for the 20 highest priority chemicals. I just pulled it from this website right here. These are the cast numbers of those 20 chemicals, high priority, and these are the predictions. These are their ranges. So that's also very important because no in vivo data, as Nicole mentioned, is as precise as one value. It's always a range, and yeah, I guess that's it. Anything else would be for the questions, I guess.
Thank you so much, Kamel. We have some questions coming in, and I think they're going to be for both of you probably, so I'll let you both answer as you like. The first one is about reporting or output, and so someone is wondering if there's a formal report that comes out that can be shared with a regulator, for example. So is there a way to kind of create a data report, I guess?
Nicole, do you want to get that? Or
well, so I mean, at the moment, not really. So when you're looking at the Opera GUI, right, the output file is the file that we've been kind of, actually, if you could let Kamel keep sharing his screen, Amy, I think that would probably be useful. So the output file is that Excel file, right? So that's the current version of the output file. But as Kamel noted, on the EPA chemistry dashboard, there is, a different type of output where you can actually look at the model results and you can get this overall picture of the performance of that particular model, the training set, the test set, the nearest neighbors of your chemical in question. And so you can print that as a PDF. Now, I can't speak to, whether or not that would fulfill criteria for EPA as sort of part of a regulatory submission package, but I think it's a fabulous idea that we could work with EPA and OPPT in particular to understand what they would like to see as far as a report output from CATMoS and make that, an option in subsequent versions.
Yeah, so the European ECHA, they have their own reporting format, and it can be, there is an app, I think, that you can download and populate, and then it will generate the report. But as far as the EPA, I think, yeah, as Nicole mentioned, this is the only report I'm aware of. And I don't think it's for like regulatory reporting, but it's just for information, I think.
Okay.
This is Gino. Can I say something?
Yeah,
Hello?
of course.
Oh, okay. I wasn't sure if I unmuted. So thank you, Jamal. Just answering that particular question. I did have a question about the European output application. I mean, I think many of you know about the OECD harmonized templates and the move towards developing these templates for a variety of endpoints and new approach methods. So I don't know, is the European using that as their output format for a CATMoS output? Are they using the OECD, or at least a proposed one? Or I know some of them are in different stages of play.
So Chris, you may be also able to speak to this, but at the moment we have really been focused on U.S. applications for CATMoS. I mean, this is all like hot off the press kind of stuff, right? We've been just working with ICVAM and ICVAM partner agencies over the last 2 years to really get this up to production level and have it be ready for use and evaluate its the scientific confidence in it for regulatory purposes, in particular for the EPA. So we haven't really gone down that road of trying to see whether or not this would be applicable to sort of international regulatory decision making. We do actually have open dialogues with ECHA and EFSA, in particular ECHA, to help evaluate the performance of CATMoS on chemicals that they care about as well. But in terms of like a harmonized reporting template, we haven't gotten there yet. But again, it's something that I'd be very interested in spending some time on in the future.
Yeah, I mean, I think that'd be interesting to take a look at how we can match them up, the outputs from a model like this with the OECD templates. We have another question about the applicability. How well does this software work with polymers or cationic substances?
So it all depends on the model. So it's per model. So if it's like a big polymer, I don't think we have any of those in the training set of any of these models. But like the repeating compartment of the polymer itself, it might be one of the chemicals, or at least be very similar to one of the chemicals in the training set. And then for those, you will have a prediction, and it will be within the applicability domain. I don't have any examples like right now, but if anyone is interested in predicting polymers, they need to enter just the repeating part of the polymer, just the main structure without an. It has to be a defined structure for it to work.
That's helpful. Great. Sort of a two-part question. One of the parts is we've found that LD50 predictions by Opera and TEST, T-E-S-T, are comparable. Have you found that Opera's LD50 predictions are comparable to those from commercial QSAR programs? And also, this person is wondering about the acceptance of data from Opera by US federal agencies.
I don't know. I mean, Kamel can comment on the comparability to other softwares. I don't think that's a surprise, especially considering that some of those softwares that were mentioned specifically were input models into CATMoS, for example. So I would certainly expect for chemicals that are inside the applicability domain of individual models or individual softwares, that would be consistent with the CATMoS consensus models. I think the point being that when you then combine lots of individual models, you can really expand out that applicability domain and provide coverage of pretty much the entire chemical universe, because you're using a lot of different approaches and a lot of different feature sets. So that's one of the real advantages of taking this crowdsourcing, consensus building type approach, and that's reflected in the graph here. And then in terms of the acceptance of these types of predictions by regulatory agencies, we're working, as I mentioned, with our federal partners to try and help evaluate the predictive performance within specific chemical spaces and domains of interest to those regulatory authorities. So we have a project ongoing with EFED right now with the eco-toxicology group in the Office of Pesticide Programs to evaluate CATMoS predictions as surrogates for acute LD50 in ecological risk assessments. So there's, a number of ongoing projects to try and help answer that question about acceptability for regulatory decision making.
Thank you. Another question. Regarding the weight of evidence approach and adjusting the LD50 based on inherent variability, if you have an LD50 range, is it reasonable to assume plus or minus 1 GHS category?
So just one thing about that. I want to mention that it's not always the LD50 value that is adjusted based on the weight of evidence approach. So when Nicole earlier mentioned it, she mentioned, so this was just one example. So other examples might be that the LD50 prediction is right here, but the GHS is right here. So then the GHS will be adjusted. And most of the cases, I noticed it's maybe around 90 or more percent that they all agree from the first time that they don't need any adjustment. So LD50 here was just an example.
Thanks, Kamel. And I'll just add on to that, I think the quantitative confidence interval that we derived from the variability analysis of plus or minus 0.3 log, it's being refined a little bit as we sort of redo the analysis while we're writing up the paper, but it seems to be pretty consistent. Interestingly, not just within this data set, but actually in other work that's come out of other groups, like work that Katie Paul Friedman has led at EPA Center for Computational Toxicology and Exposure, looking at things like repeat dose studies and the consistency or the reproducibility of points of departure in those types of studies when they're independently performed on the same chemicals. That 0.3 to about a half log confidence interval, it seems to be pretty consistent in terms of the inherent variability in whole organism animal studies. Now, it's hard to generalize that and say that it's, plus or minus a GHS category, because at the end of the day, the GHS categories span different ranges, right? Mm-hmm. So, a category 1 only covers, a certain dose range versus a category 3 covers a much larger dose range. And so I think it's better to sort of look at it more from the quantitative confidence interval. And in some cases, for a particular chemical, when you add that confidence interval, you are going to span multiple GHS categories. But in other cases, you might still be just within one category, even with that confidence interval incorporated.
Mm-hmm. A question here about the prediction file. It says in the experimental and predicted LD50 of the nearest neighbors, or some of them, are very different for some chemicals. Can you comment on how best to understand this discrepancy?
Yeah, sure. So the experimental and predictions, so you only get the range for the chemicals that you are predicting. But for the nearest neighbors, you only get, if you want the range, for example, of that predicted chemical and its nearest neighbors, then you have to take that CAS or that structure and run the prediction. But just in a second, I'll open one of the files. And so, for example, this file with the examples, so you have for each nearest neighbor the experimental and the predicted. And yes, the predictions are sometimes a little bit different from the experimental, and we notice this also when we try to interpret those predictions based on the structures. So sometimes very similar compounds have like the nearest neighbors, I won't find any example here. Like, for example, these two here, they're nearest neighbors. They're very similar. And in the data, one of them is 113 and one is more than 5,000. And sometimes it could be like an issue in the experimental values, or sometimes it's not. So the predicted for those two will be based on the structure itself and the similarity to those nearest neighbors.
That specific example is actually kind of an interesting one, because that is thalidomide. So that source, no, it's the one below it. There you go. That source compound is actually thalidomide. The closest nearest neighbor is beta thalidomide, which is almost identical structurally. And
yeah.
Thalidomide is it's obviously one of the most well-known human teratogens, so it's highly developmentally toxic, but it's not highly acutely toxic. It's used as a pharmaceutical by lots of different people, in particular for treating things like leprosy or treating cancer. It's actually a chemotherapeutic due to its anti-angiogenic properties. So Kamel and I were actually looking into this yesterday, because, obviously we're always interested when there's like huge discrepancies between what's reported as an experimental value and what's predicted by the model. And so, this is beta thalidomide. It has an experimental value of 8,000 and a very consistent predicted value. This is thalidomide, which supposedly has an experimental value of 113 mg per kg. That was, that's hard to imagine for a pharmaceutical chemical that's pretty widely used. So I don't know. I don't know what the source of that experimental value is, and whether or not it's reliable. But one thing that we did notice, and one really interesting utility of this type of model, is actually for finding annotation errors in data sets. So Kamel, if you go to the slides, you know which slide I'm talking about. So when we looked at, like, for example, the ECHA data, so we were finding, obviously one of the things that we're most concerned about is, you want to switch to presenter view. There you go.
Oh.
Go to the previous slide. So one of the things that we're most concerned about is we don't want to under predict highly toxic chemicals, right? So we, we got a set of data from ECHA that were, not involved in the model building. And so we used it as another external validation set sort of after the project was kind of completed. It came on board later. And so we were, we really wanted to make sure that we got all the category 1s correct. And so we got this chemical that, in the original data set, it said that it had an LD50 of 1.65 milligrams per kilogram per day. So highly toxic chemical. And this was predicted consistently across all the CATMoS models as a non-toxic chemical. Not totally non-toxic, but it was, it was predicted as, what was the prediction, Kamel? It was about 1,000, about something like that.
Sorry, here you have it in log, but it's 318, and that's over 5,000.
Okay, right. So it was predicted as a non-toxic chemical. And so we thought, well, this is a huge discrepancy. And so then we went back into the REACH database and started looking at this chemical. And as it turns out, it was a transcription error. So it was actually 1.65 grams per kilogram, not milligrams per kilogram. But when it was extracted from the database, it was extracted in units and assigned units of milligrams per kilogram. And we actually found multiple instances where the model helped us identify human error, or not necessarily human errors, but transcription errors in the data set that were, elucidated or revealed by the discrepancies with the predictions. So that's our, I think, a fascinating application of this type of work that we're just starting to investigate.
And always good to be able to try to follow that up to make sure that the predictive capacity is really being truthfully demonstrated, right? Yeah.
Right.
So I must apologize. We have a couple more questions, but we have really run out of time. I can offer to try to follow up on some of the remaining questions after the webinar. I just want to close with a really compelling question. Opera's results seem very robust. Is there any reason for industry to keep running in vivo LD50 tests?
You want my personal opinion on that? I don't know. Go talk to your friendly regulator.
Sounds like a plan. Well, thank you so much to both of you, Nicole and Kamel, for your presentations and demonstration. Thanks to our co-hosts, PETA Science Consortium and EPA. Thanks to all of you for joining today's webinar, and keep an eye out for future webinars, and enjoy the rest of your day. And I'll say goodbye. Thanks again.
Thanks, everyone.
Thank you.